Both Larry and I are honored to be presenting here at the Chen Symposium, and we're honored to present this individual's work, Cheng Liu. Neither Larry nor I did any of the experiments you will see today. Cheng did all of them, and you will see, I think, that he did an expansive body of work with far-reaching conclusions that's really exciting for Larry and I to be able to share with you these results. And we hope this experiment of experimentalist and theorist joint lecture works. Let's see how it goes. So today's lecture will be about navigation. And one issue when we're navigating the world is that sometimes our heads are pointed in the direction we are traveling in. And sometimes we're looking some other direction as we walk forward. And with head movements, our gaze can shift plus or minus 90 degrees, say, as humans. But in certain circumstances, our heads might be pointed 180 degrees offset from the traveling direction in the station wagon for many hours. And in all these conditions, we have an internal sense of where we're going in the world. And we can do it even though our head angle and our traveling angles might be very, very offset. And the reason this distinction is important, one reason is that the neurons we know about that indicate the orientation of our body with reference to external coordinates in neuroscience at the cellular level are head direction cells that have been discovered in many species, which indicate which way our heads are oriented, or an animal's head is oriented in an environment. But as these examples indicate, what the animal often cares about is which way it's traveling in the environment. So are there signals related to traveling angle in the navigational centers of the nervous system? And if so, how are they constructed? And this is such a general question that we don't need to go to the human brain, but we can study this in an insect. So Drosophila, with its powerful tools for elucidating biological mechanism, also makes head movements plus or minus 20 degrees, and it flies. And when you navigate through fluid media, you have to deal with currents. And sometimes those currents will take you 180 degrees offset, or any which angle offset from your heading angle. And in Drosophila, over the past 6 years or so, work from many labs, including Michael Dickinson's lab, Vivek Jarman's lab, Rachel Wilson's lab, my lab, and many others, have studied a heading signal in the fly brain. And what I wanted to tell you about today is the discovery of a traveling signal and how it's built. So let's start going into the brain, and specifically the part of the brain that carries these navigational signals, which is the central complex. The central complex of Drosophila consists of 4 principal structures. One looks like this bicycle handlebar-shaped structure called the protocerebral bridge. If you go a little deeper in the brain, you find a structure called the fan-shaped body. Beneath the fan-shaped body is a donut-shaped structure called the ellipsoid body. And under the ellipsoid body are these sets of noduli. And today's talk will include descriptions of signals in all 4 of these structures: the bridge, the fan-shaped body, the circular ellipsoid body, and the noduli. And all of these structures, one of the things they do is have a angular representation of space, or signals that cover tile angular space. And in the ellipsoid body, it's very natural. The 0 to 360 degrees around the fly are mapped around the circular structure. The bridge has 2 circles that have been opened up and pieced together, as shown here. And the fan-shaped body has a 4th axis, angular axis. All of them cover angular space. And so let's start with the bridge and the ellipsoid body. In these structures are, well, they're made of neurons, many hundreds of neurons. And here's one class of them called EPG neurons that I will always show in blue, and Larry will always show in blue. And EPG neurons have dendrites that tile the ellipsoid body, and their axons then tile the protocerebral bridge. And a key step forward in this field was done by Johannes Seelig in Vivek Jarman's lab when he imaged calcium in the dendrites of the EPG neurons in a fly that's walking on a little floating ball. And what they saw was that there was a little bump of calcium activity that was acting like a little compass needle. And so when the fly was stationary, this bump was stationary. And when the fly turned on the ball, and in this case, it's in complete darkness, this little bump of activity turned in the brain like a compass needle. So I'll play a video. The fly turns, the compass needle turns, the fly stable, the bump is stable. Fly turns, it updates as follows. And we've imaged from these same neurons in their axon terminals in the protocerebral bridge. And what you see in the bridge are two bumps of activity, one on the left bridge and one on the right bridge, respecting the two open circles that I told you about a little earlier. And these bumps move like little windshield wipers in the bridge as the fly turns on the ball. And one thing that's a little different in the experiment I'm about to show you a video of here is that we gave the fly a visual beacon to know its orientation rather than the complete darkness measurement that I showed you in the prior video. And when you give the fly a visual beacon that's yoked to the ball, so when the fly turns, this blue bar turns with the fly and with the ball, it acts like the sun or a distant mountain, and the fly can use it and the brain can use it to estimate the fly's orientation. So what you'll see in the video is when the fly turns on the ball, this bar rotates with the ball, and these bumps track the angular position of the bar, like little windshield wipers. And these compass signals in the brain, you can study them in a walking fly, or you can study them, as I'm showing here, in a tethered flying fly, which is the preparation Michael and Dickinson and I developed in his lab. And when you do this, you can track the wings instead of, there's no ball, so you track the wings, and you can have a little visual stimulus that then moves in closed loop with the wings, just like with the ball, analogously. And most of the experiments I'll tell you about today were in tethered flying flies. So the EPG neurons have this more detailed anatomy. A single neuron has dendrites in this wedge, say, of the ellipsoid body and goes up and projects to this glomerulus of the left bridge. The neighboring EPG goes to the right bridge, left bridge, right bridge, left bridge, right bridge, as follows. And so now I hope it's clear why a single bump in the dendrites of the ellipsoid body EPG dendrites manifests as two separate bumps in the bridge. It's the same exact neurons that are active in the two structures. And the EPG neurons fill up the two structures. And in fact, all the neurons we will tell you about today inside the central complex are columnar neurons in this way, meaning they tile. They have many constituent elements that tile the structures that they innervate. And so if a fly is standing at some angle relative to the sun, these bumps will be at some location in the brain. And when the fly turns with reference to the sun, that's what we're mimicking with that blue bar, these bumps will rotate in the brain. And if the fly turns 180, the bumps might go to the bottom. And at this location, you can see that these are the, when the bump kind of opens up, shows that it's an open circle because it's on two sides of the bridge. And then when it keeps rotating, the whole system rotates around. So these are bumps of activity that we think inside the fly's brain indicates which way am I oriented relative to the sun or some external cue. Another way of saying that is a world referenced orientation signal or an allocentric. World referenced is allocentric. So the question is, for this talk, what about if the fly is traveling in a direction that's not the direction it's heading? Is there a bump in the ellipsoid body or elsewhere in the central complex that says which way the fly is moving rather than which way the fly's head is oriented? And so the experiments that kind of revealed this to Chang are shown here. Here's one experiment where we are imaging the calcium signal of the EPG neurons in the ellipsoid body. You cut the ellipsoid body open into a line. You see the bump kind of moves up and down on this plot over time. That's the bump rotating around the circle, plotted as shown. And you can extract this peak of this activity, and that's shown here in blue over time. And you can plot on the same plot the position of the bar in closed loop, and you can see that this bump in the brain is following the position of the dot in this case, in this flying experiment. So this dot is rotating in closed loop with the fly's turning behavior. So here it is. Here's this signal that's indicating which way the fly is oriented relative to this dot. And the question is there a signal that indicates which way the fly is traveling relative to the dot? Now, the fly is glued to a plate, so it's not actually traveling anywhere, but it might be trying to. And so what Chung was doing in this experiment was actually co-imaging this blue bump with other bumps. And he did a series of experiments where he imaged a bunch of other signals in the central complex. And there's many other columnar neurons that have bumps of activity. And one of these other neurons that he imaged are tile the fan-shaped body. They're called PFR neurons. And they also express a bump of activity that moves left and right in the fan-shaped body. And so Chung was co-imaging these neurons with the EPG neurons in the experiment I just showed you. And what he saw was interesting in that the bump in the PFR neurons was broader, first of all. And sometimes, like here, this broad bump follows the arc of the blue bump. You can see that the peaks are tracking together. But at other times, like here, the blue bump was relatively stable and the purple bump is whizzed across the fan-shaped body, as shown here. So there's a bump that sometimes is aligned with the EPG bump and sometimes isn't. And so that suggested to us that maybe the reason it's misaligned at times is because the fly is trying to fly sideways or backwards, and this purple bump is indicating that. And so the way we tested this in the flight experiments is, if you look in this arena, sometimes we present it to the fly's optic flow, so a sea of dots beneath the fly. So it's still controlling the closed loop sun stimulus, but we presented this optic flow to make the fly feel like it's getting blown forward, in this case of optic flow, or backwards. And this is very compelling if you've ever been in a virtual reality bubble or glasses on, goggles on. For humans, it's very compelling, and we think the fly is finding compelling too. You see the sea of dots that make you feel like you're moving one direction or another relative to your body axis. Okay, so in parallel with this closed loop sun, we presented this optic flow. And I just want to mention that this optic flow stimulus was adapted from code from Michael Dickinson's lab. And in fact, Peter Weir, who was a postdoc with Michael Dickinson, was the first to image fan-shaped body neurons in response to these optic flow stimuli. And what we saw in these experiments was that when we presented a static optic flow, that's what I was showing to you here. Now, when we presented optic flow that simulated the fly's body moving forward, we saw a remarkable and very strong effect, which is that the fly could still turn left and right, and the sun and the bump position here indicated that. It locked into the position of the dot. But now the purple and blue bumps aligned very tightly. So when the fly's body was traveling in the direction it was oriented, these bumps aligned. And when you didn't give the fly a cue that it's moving forward, they were misaligned again. So we could do this sort of experiment not just anecdotally, but systematically. Here's another single example when we presented for 3 seconds optic flow forward. And you can see that in this sample trial, the blue and purple bumps aligned. And this is now 13 flies. The difference between the blue and purple curves over time, and they are not very different when you present open loop optic flow simulating forward travel. When you simulate backward travel, they got misaligned by exactly 180 degrees. And when you presented the intermediate drift angles, you got intermediate offsets that were very consistent across individuals. And you could summarize these data by plotting the difference between the blue and purple curves as a function of the optic flow angle. And these fit decently along a linear axis, which means the difference between these two faces is saying which way my body is getting blown. And kind of maybe even more precisely, more relevant to our question is you could infer which way the fly's body is moving in a world-centered coordinates from these measurements. So you could say, where is the sun in the arena, or where's this blue phase? You could say that's the way the body is oriented. And then you could add at every moment in time, you could say which way the optic flow is moving and add that angle. And so from those two angles, you can say which way the fly's body is moving relative to the world and plot that on the x-axis. And you can plot on the y-axis, where's the purple bump in the fan-shaped body? And then you see a very nice, beautiful fit. The position of this purple bump is closely tracking which way the fly's body is moving in a world-centered reference frame. And we saw this not only in PFR neurons, but in the input. They have a dominant input from a set of neurons called H delta Bs that we will always plot in red. H delta B neurons also have this traveling direction signal. And in fact, it is in H delta B neurons that we think this signal is first built. And so let me summarize what I just said in a cartoon. If you didn't follow the data, this is the point. When a fly is flying, there's two bumps of activity, one in the fan-shaped body, one in the ellipsoid body. Both these bumps indicate an orientation relative to the sun or relative to the world. So literally they represent east, north, west, south, and so on. And the reason we don't use these terms usually is because in any given fly, east might be here in the fan-shaped body, or it might be here. But once you get this offset dealt with, which could differ fly to fly, these are literally Earth coordinate systems like north, east, west, south. So when the fly is flying around, the bumps track which way the fly is oriented, but when the fly gets blown backwards, right there, the purple bump says I'm moving south, and the blue one doesn't. And now the fly gets blown back again. The purple bump is saying which way am I traveling in the world, even if it's not oriented with my heading, and the blue bump doesn't. And so that's the signal we found, and that's the first part of the talk. And now the rest of the talk will be regarding this question: how do you build a bump of activity in the brain that tracks which way you're traveling relative to the world, or an allocentric traveling direction signal? And with that, I'll hand over to Larry.
Gaby introduced to you these two directions, the heading direction and the traveling direction. And I want to outline how the traveling direction would be computed, just the components we need. We'll go into more detail as we progress in order to compute this traveling direction. And an important feature, which Gaby presented to you, is its traveling direction in the world, reference to the world around it. So the first thing that is going to be needed in order to calculate that is a reference to an external landmark like the sun here through this EPG compass system. So in fact, we think that a lot of the computations being done in the central complex are probably tied into the world around the fly through this system. And that's definitely true of this traveling direction signal. So that links it to the world. But of course, the fly also is going to need to know which way it's moving in the world. And that's done through optic flow. As we progress, Gaby will explain to you how visual neurons project the optic flow onto these four directions that I've shown in brown and orange. There's an orthogonal system that's rotated 45 degrees relative to the fly, and the optic motion is projected onto those vectors, as I said. The key in linking these two, the right-hand side would give you an egocentric view of motion. The left-hand side ties it to the world, is to tie these four axes to the heading direction. In other words, the front two there, you wouldn't call plus and minus 45 degrees, but heading plus and minus 45 degrees. Then when the fly rotates, they get rotated along with it, and they're constantly referenced to the world. And that is how the traveling direction signal is computed as a vector sum. Once you do this first step of anchoring these vectors to the world, that's the coordinate transformation from egocentric to allocentric. And then it's a vector summation problem, which I'll show you now. So if you take these four vectors for any particular drift or optic flow, this would be a little optic flow that's more in the forward direction, a little bit more on the right. Now you just add these vectors and you get the traveling direction in the world. I used to teach first-year physics. This is a classic first-year physics problem, and there's the answer that a good first-year physics student will give you. This angle is given by this formula. Now, if you've forgotten your first year's physics, don't worry, because we're going to explain to you how the fly calculates this angle. It's exactly the same calculation, but it's actually done in a clever and much simpler way. There's no ratio, there's no arctangent, and so we're going to progress to tell you how the fly does this computation. The first question that we have to ask experimentally then is how and where are these four vectors represented in the fly's brain?
So the answer begins with the fact that the EPG cells, when they project up to the bridge, connect up to a set of neurons that are called PFND cells and PFNV cells. And this anatomy of these cells was first described by Tanya Wolff in 2014, and has been kind of even in more detail described with the recent release of the connectome from Genelia, which will serve a role in kind of anchoring our anatomy here. So I'll be telling you about connections that are analyzable in that beautiful data set that Genelia released. So the EPGs we know now through those two sets of experiments connect, or papers, connect to these PFND and PFNV cells directly. And when you co-image, when Chung co-imaged the EPG cells in a green fluorescent calcium indicator and the PFNV cells in the bridge with a red fluorescent indicator, he saw a very clear result, which is that the PFNV, these orange cells, have 2 bumps of activity, just like the EPGs, and those bumps track the EPG bumps kind of in a relatively boring way. So in addition to the 2 bumps of EPGs moving like windshield wipers, we have 4 more bumps in duplicate, 2 PFND bumps and 2 PFNV bumps. So those, we believe, might get us part of the way there towards these 4 vectors. The reason we think they might be related to these 4 vectors that would be building this traveling signal is twofold. One is these 4 things are anchored to the heading angle that Larry talked about. When the blue bumps rotate, all these orange and brown bumps will rotate together. So they have that H- calculation in there for free. And the second thing is, as we'll show you later, is they connect monosynaptically to these H delta B neurons that we know have the traveling signal in them. And so we have 4 signals that are connecting to our traveling bump neurons. And so they have the potential to carry these 4 vectors. And so can bumps, they don't look like little arrows in the brain, they look like little bumps of activity. So our next question is going to be, can bumps represent vectors?
So the answer to this is yes, if they're the right kind of bumps. So there's a very well-known representation of vectors called a phasor representation, in which you take a vector like this, has a length L1 and it has an angle phi1, and you map it onto a sine wave. And that sine wave should have a phase equal to the angle of the vector and an amplitude equal to the length of the vector. So this is the phasor map between sinusoids and vectors. You can do this for a second vector. It has a length L2 and that maps to the amplitude, and the phase maps to the angle again. And then the payoff here is that if you want to add those 2 vectors, which you can do by the vector method, all you have to do is add the 2 sinusoids. So you add the 2 sinusoids, you'll get a resultant sinusoid that has its phase at the angle of the summed vector and its amplitude equal to the length of the summed vector. So in engineering, this trick is used on the right to sum sine waves using vectors. And what we're going to argue is that the fly does it the other way. It uses the sinusoids to add the vectors rather than the vectors to add the sinusoids. Now, the idea here then is that these are spatial sinusoids, not temporal, that populations of different cell types, like you see on the bottom there, through their activity across the structures of the central complex, are going to map out these sinusoids. And we'll have 1 for each vector we need. They'll get summed because they'll converge onto a common target. And that's the proposal for how things work. Now, this idea has been noted before. There are papers that go back to proposing that maybe something like this is going on in the hippocampus associated with various navigational computations. There are also that's the first two papers that I referenced there. The last 3 papers actually apply this idea to insects and to navigation in insects. In particular, the last paper here, the Stone, Webb, and Heinze, use these ideas in an interesting proposal about how path integration might be done in bees. And it also used an analogous circuit in bees, quite similar to the one we're describing. So the question that we have to ask now of the experiments is not just that there are PFN bumps, but are they sinusoids? Because that's critical to this idea.
So the way you can test this is by taking the bumps, in this case in the EPG measurements, phase nulling them, meaning move the bumps so they're always in the same position on every frame, and then average the GCAM signals that you see, the calcium signals, and look at the shape that you get out. And when you do that with EPG bumps, these blue bumps, you can see a variety of shapes, but often you'll see a shape that is sub-sinusoidal, as shown here. And with a sinusoidal fit shown in a dotted line. And you can see it's not very good. And this shape of the two bumps in the bridge makes sense because if you project this bridge through the known anatomy down to the ellipsoid body, a beautiful kind of Gaussian looking bump in the ellipsoid body would yield this, because every other signal goes left bridge, right bridge, left bridge, right bridge. You get this half bump on the left, half bump on the right, and they're kind of too skinny often. So the EPG bumps are in many measurements not sinusoidal. What about the PFN and D and V? It turns out that they're comport really, really nicely to sinusoids as measured in the bridge. And not only that, the kind of the beauty of the Drosophila system is that you can imagine a hypothesis rooted in detailed anatomy for how you convert a non-sinusoidal pair of bumps into a sinusoidal pair. And the idea is this, that when the EPGs go up to the bridge, they talk directly to the PFND and PFNV cells, but they also talk indirectly to those same cells through these interneurons. So EPG cells hit these delta 7 cells very strongly with their synapses. And here's one example of a delta 7 neuron that has this kind of weird and beautiful anatomy. It has dendrites that peak at these two positions, and you can see kind of anatomically drop off in their intensity off their peaks. And these three locations are where the axon terminals are of the cell. So let me draw this cell schematically on our bridge here. And you can see that when the bump is at the top of the ellipsoid body, which is the kind of situation we're going to be simulating, this specific delta 7 green cell is going to be activated very hard if you assume that anatomical overlap co-varies with physiological activation. So this green cell should be activated very strongly, which means the outputs of this cell should be very high to its downstream targets. And so we can draw this kind of schematically in a simple model where we schematize the dendritic weights as varying, going up and down sinusoidally twice over the bridge. That's shown here, up and down twice. And then the same cell's axon has outputs here, here, and here. So I've modeled the single cell as its dendrites here and its axons here. And in this model, you can take this average activity from the EPG, take a dot product with this simulated dendritic density, and that's just multiply point by point and add everything up. And you can see the peaks are aligned, so you're going to get a large value. It's going to give you a nice output. And so these three locations are going to be very active. And the key is that this is just one of 8 different delta 7 neurons. The next delta 7 neuron has everything shifted over to the left. So if you look at this schematic, here's another delta 7 neuron with its axons here. Now this blue neuron is going to hit a little offset from the peak, so the output at these two locations is going to be not quite as high, but still high. And another delta 7 neuron has its axons here. It's going to be, its dendrites are very thin here. So this cell would be very inactive. So you can plot all 8 delta 7 model neurons, all 8 modeled outputs. And what you find in this simple model is that if you take this weird EPG shaped thing, play it through the model, you get a beautiful sinusoid out. A couple things to say. The sinusoid is phase inverted, so this is the trough where you see the peak here, but we're almost certain that the delta 7 neurons are inhibitory because they're glutamatergic, and there's functional evidence as well to suggest that they're inhibitory on many downstream cells. So what we think is happening is that the EPGs hit their downstream cells, and these inhibitory reshaping neurons kind of reformat these weird bumps into sinusoids. This same idea is in analysis of the connectome paper from Vivek Jaraman's lab as analyzed by Dan Turner Evans. And so just to summarize schematically, you take weird shaped bumps and you can turn them into sinusoids. How do you do it? You get direct inputs to the cells and you have indirect through delta 7, and we think these delta 7s reshape these 4 bumps to be 4 sinusoids. And so we have 4 sine waves now in the bridge that have an interesting property. Not only are they in the bridge, they're yoked to the fly's heading. So when the fly turns, remember I told you the EPG bumps would move in the bridge like this, and these are driving these 4 sinusoids, so they're going to rotate as well. So every time the fly's turning, you have these sinusoids rotating in the bridge, which means they're yoked to the fly's internal sense of heading, And sinusoids, as Larry said, could represent vectors. So we're in good shape. And I guess I'll throw it to Larry by asking, can we declare victory? We have 4 sinusoids linked to the fly's heading.
Yeah, so we have a lot of good news here. As Gabby said, we've got our 4 sinusoids. It seems like the system puts some effort into making sure they're really sinusoids, which fits with the phasor idea. They're all linked to heading direction because they're all tied in with the EPG heading signal. So this axis I've drawn on the right is the heading angle. And I don't have a nice movie like Gabby, but they would all move together back and forth if the fly turned. So that's all great. We've made the coordinate transformation to world coordinates because all of these phases are now tied to heading, and that's what we needed with the vectors. But we've got one problem, and that is that these guys are all aligned. That means that the 4 PFN cell classes represent 4 vectors that are all parallel to each other, and that's not what we wanted. So we almost made it. But what we really want is that these 4 vectors are splayed out and differ from the heading direction by plus and minus 45 in the frontward in the PFNDs and plus and minus 135 for the PFNVs in the backward direction. So what I have to introduce then is another question about the data, which is, are the PFN sinusoids shifted in this way?
So it turns out that the 4 sinusoids in the bridge, I've drawn them up there as 2 brown bumps on the left, 2 orange on the right, are aligned with the EPG bumps in the bridge. That's another way of restating what Larry just said. In the bridge, all the bumps are aligned with the EPG bump. But the key issue is that the way the traveling direction signal is going to be built is through interactions down here in the fan-shaped body. So what we need to analyze is how these neurons project to the fan-shaped body and interact with the H delta B neurons that they synapse onto. And so that's what I'm going to show next. Let's look at these 2 single PFNV neurons, and I'm going to draw their dendrites in squares and their axon terminals in circles. They have this kind of beautiful crossover anatomy that is shown by Tanya Wolf and by the connectome released more recently. So what does this mean? This means that the fan-shaped body's axis, if you say this is 0, the 2 corresponding cells here are offset by 90 degrees, plus or minus 45 degrees from the center point. And so you have a 45-degree shift just in how they project down here. Now, these orange cells are not the 45-degree shift. These cells are going to be the 135-degree shift. And the way this works is that the H delta B cells have introduced a second shift of 180 degrees. So now let me show you what these cells schematically look like. H delta B cells have dendrites in one column of the fan-shaped body, and they have axon terminals that are offset by 180 degrees here. So if you add 45 to minus 180, you get a net shift of minus 135. And so this bump of activity gets moved and placed here in the fan-shaped body, or this neuron's activity, I should say. The symmetric process happens from the left bridge, and so we have our plus 135 and minus 135 shifts. In PFNDs, they have the same first-order anatomy as PFNVs. The difference is that they connect, PFNDs connect very weakly, relatively weakly, I should say, to the dendrites of H delta Bs. They do connect to the dendrites, but more weakly than they connect to the axon terminals. So it's kind of an interesting and odd anatomy. This PFND cell will drive this axon terminal, we believe, stronger than the dendrites of the cell here. And this PFND cell will drive this axon terminal here. And so if you look at the axons of the H delta B cells, you expect that these 4 common positions in the bridge will lead to 4 offset positions in the fan-shaped body that correspond to plus 45, minus 45, plus 135, minus 135. And this same anatomical insights are in a paper by Jenny Liu and Rachel Wilson with theoretical work by Shaul Druckmann that's on bioRxiv. So let me kind of summarize here with our sinusoid and vector diagram, which is summarizable as follows. We have these 4 bumps aligned with the EPGs in the bridge, but the way these bumps, these sinusoidal bumps, project to the fan-shaped body is with a unique anatomy. So PFNDs go down plus 45. I should say minus 45, the way we flip the axes here for technical reasons I won't get into. This, so meaning this vector gets positioned here relative to the front. This one goes to the other direction, 45. PFNVs go here, but then the H delta Bs brings the peak over to here, and vice versa for the other one. And so the 4 bumps in the bridge are sinusoids. They can represent vectors. And not only that, we think they get summed by the H delta B cells in this kind of 90-degree offset way with peaks at the exact positions Larry's has been talking about to you. So now I think we can ask, are these properties sufficient? Can we say that we can build a model for how the traveling direction signal is built?
So we've made great progress here. We've got our 4 vectors represented by sine waves. The sine waves are oriented in the right way to represent the angles of the vectors, but there's one piece missing. And that is that if you remember, when I made this phasor analogy, the phase of the sine wave corresponded to the angle of the vector. That's the part we've accounted for. But also the amplitude of the sine wave was proportional to the length of the vector. And if you remember, the lengths of these vectors are going to reflect the projection of the optic flow onto these different directions. So we've got to have a case where optic flow modulates the amplitude of these 4 sinusoids that we're talking about. Let me just illustrate you that for one of the vectors, the one corresponding to PFND on the right side. So here's sort of the null point where we've got a vector and a sine wave. If there happens to be a lot of optic flow in that particular direction, the vector gets longer, and that means the sinusoid has to get of higher amplitude if this is going to work. If when we add up these sinusoids into the H delta B, we're going to get the right answer. Likewise, if there's low optic flow in this particular direction, the sine wave should shrink. So again, we're going to come up now with a question for the experimentalists, which is, are there amplitude modulations of these sinusoidal spatial patterns in these cell types, and are they related to optic flow?
So a remarkable observation by Chung was that there are extremely strong modulations of the amplitude of these signals in the protocerebral bridge with optic flow. So here I'm showing you the mean of 9 flies when he was imaging PFNDs. We phase nulled all the signals and averaged them so we can see the shape and amplitude of the GCAM signal in the left bridge PFNDs and the right bridge PFNDs, and the same for the Vs. When we presented optic flow, in this case that simulates the fly drifting directly backwards relative to its body, the Vs became very bright and the Ds became very weak. And by the way, these dotted lines are sinusoidal fits. And so you can see that they still are sinusoids, they just change amplitude. When the fly was simulated to be going forward, the amplitude modulation inverted, where the Ds became bilaterally higher and the Vs became bilaterally weaker. And in these kind of off-center, off-axis directions, we saw these asymmetries in amplitudes. So the answer is that we saw extremely strong modulations in amplitude, very dramatic ones. And we can quantify these amplitude modulations as follows. You can take the sinusoidal fit and extract from it the amplitude of the fit, or do it directly from the data, and you can see that the amplitude of the left bridge PFNDs grows and shrinks as follows with the optic flow direction, and that's what I'm plotting here, kind of the amplitude fit. And you can do the same thing for the right bridge. You can see that it would peak right around here, and that's what I'm showing on the right. You can do the same thing for the Vs, and you see these strong modulations that themselves are sinusoidal. So this is a sinusoidal modulation of the amplitude of a sinusoid. These are just experimentally, these are not generically observed. If you look at EPGs, they're relatively stable in amplitude. And one of the things I think Larry and I want to do with our hour here that we have because of a joint talk is really show you the level of comprehensiveness we can provide to the understanding of these signals, which is like with the delta sevens, we could say, hey, maybe these neurons are contributing to reshaping bumps into sinusoidal bumps. Here, we can use the anatomy of the inputs to the central complex to give a hypothesis about where these modulations are coming from outside the central complex. What is bringing an optic flow sense from the eye into these neurons to change the amplitudes? And we think we know the answer. So the PFNVs, what we need is that the left bridge grows in amplitude and shrinks with amplitude with this property. So you need to modulate all these neurons kind of synchronously. And so the way this works is that all these left bridge neurons have another neuropil where they get input called the nod. So this is the right bridge PFNVs get inputs in the left nodulus. They all kind of converge and have a massive kind of strong input right here from a set of neurons that are called LNO2s. And so before I show you those neurons, let me just show you that when you image the PFNVs in the left nodulus and take the mean GCamp activity here, it comports really well to the mean GCamp or amplitude modulation we saw here. So let's look at the right bridge. This is now imaging in the nodulus. So this is a different set of experiments where we image these neurons down here. And you can see they have the similar modulation. So something we think that's bringing in inputs right here is driving this modulation, and that's percolating up to the bridge that's amplitude modulating the sinusoid. What are those neurons? Like I said, from the connectome and from previous light microscopy anatomy, the best candidates are these LNO1 neurons, cells that receive inputs outside the central complex and densely innervate the right bridge PFNVs in the left nodulus. When you image those neurons, you see a beautiful modulation as well. Perfectly sign inverted to the orange neurons modulation. So we are believe this is an inhibitory input, a sign inverting input that's driving these PFNVs, bringing in the optic flow input into the system. When you look at the left bridge, everything is symmetric, and you see that the LNO1s from the other side of the brain bring in an input that could create this modulation. Symmetrically, let's go to the Ds now. When you go down to the nodulus, you see the matching signals like you'd expect. And there are two neurons called LNO2s that are likely to bring in optic flow inputs to these noduli, but we were technically limited from imaging them. We didn't have a GAL4 line that allowed us to do it cleanly. Luckily, with PFNDs, they have a second input that tackles the left bridge or the right bridge kind of en masse, and they're called SPSP neurons. There's two of them, and you can image them on the left bridge and ask, are they bringing in optic flow input from outside the central complex? And the answer seems to be yes. So when you image the SPSP neurons, you find that their GCamp is modulated in a way that's perfectly sign inverted to the PFNDs. And we think that they're one of the inputs that brings in this optic flow signal into the system, modulating the amplitudes of the sinusoids so that the vector calculation can have the right amplitudes to all the vectors. And so at this point, I think Larry will be able to put it all together and give you a sense of how the model works for building a traveling direction signal inside the system.
Yeah, so let me put it all together and do that. So on the right, you're seeing these four vectors that we've been talking about all along. And in the center is a schematic of the central complex. The EPG signal here has been put in a certain place corresponding to a particular heading of the fly. And then if you go down from the bridge into the fan-shaped body, what you see is four sinusoids. Those are the spatial patterns of the four PFNs representing the forward and backward direction. And then a red dot, which would be the corresponding position of the H delta B, which sums up the input from those four sine waves, and in this case is in the corresponding position to the EPG. So this would be a case where the traveling direction and the heading direction align. But let's look what happens if the fly would start drifting backwards due to a strong wind. So in the experiments, that's represented by having a convergent pattern of dots on the screen. And Gaby showed you that the effect of that is to shrink the amplitude of the forward representing PFNDs and expand the amplitude of the PFNVs. So if you look in the middle plot, that corresponds to the dark brown sinusoids getting smaller, the orange ones getting bigger. That causes an input that drives the H delta B bump all the way across to the 180-degree position. If I look at it in terms of vectors, what's happening is the backward vectors are getting big. The frontward vectors are getting small because of the backward drift. If you add up these four vectors, the way that the sinusoidal sum is doing it, you get an H delta B signal that indicates backward traveling motion. On the other hand, if the fly moves forward, that would be represented in the experiment by diverging optic flow. In this case, things flip. The PFNDs get big amplitude modulation and the PFNBs get small. That's represented in the middle now by the orange sinusoids being big, the other ones being small. And that will, yeah, there it is. Sorry, the brown one's getting big and the orange one's getting small. That will drive the H delta B back to its original position. Now we're heading forward. If you look at the vectors, what's happened is the forward ones are big, the backward ones are small. You add them up and you get that forward direction. I'll just do one more of these. If the fly is going sideways a little bit, that would be represented in the experiment by moving the focus of the optic flow. That causes an asymmetric pattern in the PFND sinusoids. If you look in the middle, that means that two of these sinusoids on the right side will be expanded in amplitude, the other two will be shrunk. That moves the H delta B bolus to the right. And in terms of vectors, we have big vectors on the left, small vectors on the right, and they add up in that way. So this is how the system works. We can put it together into a model and compare to the data. That's what you'll see here. So on the left-hand side, the circles represent the H delta B data that Gaby showed you right at the beginning of the talk, and the plus signs are the model. You can do an analytic model here. If you notice, in Gaby's plots, all of the fits that he showed were cosines and sines of various forms. So you can do the whole calculation by trigonometry, and it just reproduces exactly the calculation that I showed you in that equation towards the beginning of the talk. On the other hand, this plot, it just uses the data. So there are no parameters in this model. You just take the data from the PFNs, you add it the way this model says it should be added, and you get those green pluses that agree very well. In the case of the PFR on the right, you'll notice again a very good match of the model, but a less good match to the diagonal. So there's this little deviation from the diagonal, and actually we understand that. That's because there is an extra input from the PFR, and when we include that in the model, we match this little deviation quite well. So I'm going to wind up my comments here, and Gaby will take it to the end. But I just want to say that for a modeler, if there is, it's just a thrill to work on a system where we've reached a level of precision that's really in system neuroscience really quite remarkable. That's due, of course, flies have always been famous for genetics, and that's only increased. But now, you know, Michael in the introduction talked about the advances in behavior in fly. You can do these virtual experiments. Now we have the EM connectome. And so I think it's kind of a new era of precision in both measurement and modeling that we've achieved that hopefully will expand to other systems over time. So I'm ready to declare victory, Gaby. I'm happy. So what do you have to say?
So maybe the last thing data-wise that was worth showing you is what took Chang about a year to do. So if Larry was able to declare victory, we wouldn't have had to do these experiments. But one of the great aspects of Drosophila, at least in the modern era, is that we can do genetically precise perturbation. So I just want to show you 4 experiments that Chang did where he tried to perturb the system in a way that would test this vector model. And the results are turned out consistent with this overall view. So the first experiment he did was to silence the EPG neurons. So that should be a major hit to the system. You don't have any anchoring signal. You can't orient any of these vectors to the external world. And so when you see the bump in the fan-shaped body, if there even is a bump, it should be all over the map. The way to represent that is that there shouldn't even be these, something very bad should be happening to these vectors. And indeed, this is now in a walking fly, he did it to be stringent, because in walking flies, we often see a nice alignment between this traveling bump and the blue bar on the screen, because flies typically walk forward. What Chung saw in the context of an EPG silence fly is that this purple bump still existed. So that's interesting. It means the system is trying to estimate the traveling direction, we think, but it's having a horrible time doing it. And you get the difference between the black and purple signals here is nearly uniform. That means the purple bump is all over the map, all over the fan-shaped body for a given bar position. So the system indeed is really reliant on this EPG starting point. Chung inhibited the PFNVs by expressing in them a potassium channel that hyperpolarizes them. That should cause in the vector model backward facing vectors to shorten, which means this should be a fly that thinks it's traveling forward all the time. And indeed, when he images a flying fly in the context of no optic flow, so this is a fly, remember, where the purple bump deviated from the blue a lot. Now they're very tightly aligned across our population, more so than on controls. So that's consistent with a fly that's believing it's flying forward. What if you excite these orange neurons? The way we're going to do it is by inhibiting those LNO2s, which inhibit, so that, disinhibition of the PFNV. So we expect this fly to have a kind of a horrible life when the light is on anyway, where it feels like it's going backward all the time. And so Chung did this with 2-photon activation of the optogenetic reagent, and that's what he's showing here. And you can see the purple and blue bumps tend to be deviated by 180 degrees. And that was true across a population. The 2 bumps deviated on average by 180. You can get the same result by shortening the front 2 vectors through optogenetic experiment. And that too caused the deviation to be on average 180 degrees. And so that's where we'll end with the experiments. I have a little bit of a conclusion to augment what Larry said beyond this, but just to say that this set of perturbation experiments is what gives us even more confidence than the models fit to the physiology. Altogether gives us a very strong belief that this is a good working view of what the system is trying to do and how it does it. All right, so 4 conclusions. I think we've shown you that flies can explicitly track the direction their body is traveling, not just the direction their head is pointing. They represent vectors, we think, explicitly, 2-dimensional vectors through sinusoidal patterns, whose amplitude represents the length of the vector and whose phase represents the angle. They can perform a rotation of vectors, scaling the length and adding them. So vector arithmetic is happening, we think, in the system. And I want to end by saying a few words to relate this work to mammalian neuroscience and our mammalian colleagues that are listening to this talk. I don't do this work, and most of the members of my lab don't do this work just to get insight into the mammalian brain. We find the majority of brains on this planet are insect brains, and so they're worth understanding in their own right. But I think there's also, in this case, relatively direct and kind of enjoyable analogy with work that both Larry and I contributed to in parietal cortex. So let me build up to that. What am I getting at? One way to think about the computation we just told you is that this system is taking optic flow input, which reflects which way my body is moving or head is moving. That's what the optic flow says. Which way is my body or head moving relative to my body? And what it's doing is converting that egocentric traveling, body-centric traveling, into a signal that's which way am I moving relative to the world, relative to the sun. And how do you do it? You integrate a sense of which way your body is moving relative to its axis with another signal that says which way your body is oriented relative to the sun or relative to the world. You combine these two, and together you can say which way am I moving relative to the world. How do you combine these two? you have a set of neurons that explicitly combines them. So PFNVs and Ds are neurons that are conjointly tuned. They have mixed selectivity. They're tuned both to allocentric heading. So basically where these peaks are of these PFNV neurons says which way am I oriented relative to the sun. And then their amplitudes indicate how fast my body is moving relative to the body axis. And so these kind of conjointly tuned neurons, what do we think they're doing? They're transforming body-centered traveling to world-centered traveling. They're performing a coordinate transformation. And what we can do in Drosophila in the central complex is image spatially these bumps of activity, and everything makes sense and it's very beautiful to see. But in mammalian systems, what often people do is you image or record from single neurons that are salt and pepper in the brain. And I just want to say a few things that commonly when you look at the mammalian brain and you record from single neurons, you'll find cosine tuning to properties. And so one thing the mammalian colleagues, I believe, should be thinking about is, are these potentially two-dimensional vector representing neurons? Are they operating in a population of cells that are not spatially distributed as nicely as in Drosophila, but exist as a vector representation? So that's one comment. Cosine tuning might reflect that sort of function, and we should consider it when we record in the mammalian brain. And the second comment I want to say is when you record in cortex, it's more the rule than the exception that you see mixed selectivity for features. So when you record from single neurons, if in the fly, had we done single neuron recordings, we would have found these PFN cells are sensitive both to heading, and that heading is scaled, the activity is scaled by the optic flow. That's analogous very nicely to work from Richard Anderson and modeled by David Zipser in the 80s, where they recorded in parietal cortex neurons from area 7A. And what they saw in those single neurons is that they had receptive fields that were yoked to the retina, normal receptive fields, position of stimulus on retina. But if the eye was in a different part of the eye socket, that response was scaled multiplicatively. And presciently, in a beautiful model, they imagined, oh, maybe these neurons, what they're doing is converting stimulus position on retina to stimulus position relative to the head. They're performing a coordinate transformation in this model. And what I think we can add to this picture is that this three-layer network idea in a computational model, initially by Zipser and Anderson, but also by others, including one of our speakers in this seminar, I think in Drosophila, we have very strong evidence that that is indeed what happens. There's a three-layer network that's doing a coordinate transformation. And to Chung's credit, he took 8 cell types and has first-order functional descriptions of what roles they serve in that transformation. And that's a testament, I think, to Drosophila and what we can do at a systems level of understanding, as Larry was saying. And it's what might come out when we, understand these circuits at that level of resolution. But isn't it remarkable, personally, and I think for Larry as well, that we had spent time in cortex and that anything we would do in the fly would bear any relation. And the idea that the higher brain functions of the fly might be performing a coordinate transformation as one of their dominant functions. And that being a hypothetical function for not just parietal cortex, but people think in the medial entorhinal hippocampal system, in prefrontal cortex, there's a lot of conceptions that mixed selectivity might serve this role of coordinate transformations. And so I want to propose that in Drosophila, we can tell you for sure that a mixed selectivity neuron is performing that function, we believe, and maybe re-energize that view of the higher brain regions of mammals as well, is one speculative hypothesis. And with that, I'll end and say you can read about this work in a bioRxiv paper we put out. A parallel study from Rachel Wilson and Jenny Liu is also on bioRxiv on many of the same results and ideas. And the vector addition ideas and then the sinusoidal ideas as analyzed in the connectome is in a paper or closer to a book, enormous tome of a very extensive analysis of the fly connectome, the central complex connectome from Vivek Jaraman's lab, and these 4 senior authors. And with that, I'll end. Thank Chung for all his work. Thank you for listening. We'll be happy to take questions.